So the first thing is that we have coined this new term, Socio Microbiology, and Vittorio alluded to this morning. When I started out in microbiology, we didn't think of bacteria as social creatures, and now we do. And we can study their sociobiology just like we would study the social activities of other organisms. And sociomicrobiology can be considered a subdiscipline of microbiology. It's also a subdiscipline of a field called sociobiology, and that's the study of the genetic and environmental influences on social activities of organisms. And sociomicrobiology has become increasingly important in the field of sociobiology. So now we understand that bacteria are really important tools for studying the costs and benefits of social behavior. So why do we study sociomicrobiology? Well, there are several reasons. Some that come to my mind, as I said, bacteria are wonderful models to understand the biology of sociality. I like bacteria, and if you want to understand bacteria, you can't ignore their social activities. It turns out that the type of quorum sensing that I work on has provided important tools for synthetic biology. The regulatory systems can be used to control genes in ways that synthetic biologists find useful. And there's been this idea, I don't know if it's true, that we can develop new medicines to treat devastating bacterial infections by focusing on social activities of bacteria. And then I have this. So this is the front cover of an issue of Wired magazine that everybody a lot younger than I am reads. And on the back inside cover, they always put somebody's prediction of the future. And about a year ago, this was the prediction of the future. This is a little handheld device that's an electronic medical bill. So all of these things are billings, and you press some buttons here and it extracts money from your bank account and pays your medical bills. And if you focus in on this one right here, this was a charge for quorum disruption antibiotics for vancomycin-resistant staphylococcus. So this is somebody's idea of the future. And I was delighted to see that this was one of the more expensive charges. So today what I want to do is quickly review some general thoughts about quorum sensing and then talk about quorum sensing and Pseudomonas aeruginosa. And the way that I'll develop this story is I want to talk about what we call an orphan signal receptor, and Vittorio calls a solo. And everybody will call it probably these things lots of different things during the 2.5 days of the course. I want to tell you about QSCR, and QSC stands for Quorum Sensing Control. And this was work done by these three individuals, Sudha Chugani, who's still in my laboratory, and she's a senior research scientist, Yannick Lequette and Junhee Lee, who were postdocs a few years ago. And here they are. And then this will lead into a story about LAS-R, because I'll pose the question, why does Pseudomonas have QSCR? LAS-R is on top of the quorum signaling cascade in Pseudomonas. And this was work that was done by Kaya Sappington, a graduate student in the lab, Ajay Dandekar, and Kenichi Oinuma. Ajay is a pulmonary fellow, and Kenichi, until last month, was a postdoctoral. He's from Japan, and he chose to move back to Japan, it turns out, 2 weeks after the earthquake. Unbeknownst to him, that was coming. And here they are, and Kenichi's back in Japan. So what is quorum sensing? seems like the beginning of this meeting would be a good place for someone to define it, and so I thought I would. It's a type of cell-cell signaling used to control specific genes, and the genes are different in different bacteria, and it allows coordination of group activities. And I've underlined group activities here because when a group is working in a coordinated fashion, that implies there's some sort of sociality. And the system that's most well studied is the type that Steve Ferrand introduced to you, Acyl HSL, quorum sensing. This occurs in the proteobacteria, in many proteobacteria. There are now over 200 species that use Acyl HSLs. And an important point here is that the signals are dedicated. They're made by the so-called Luxi family of signal synthases, and there are co-evolved receptors, the Luxar family of receptors. And this is an important point because people that work on communication tell me that for a signaling system to be a form of communication, the signals and the receptors have to have co-evolved. So this seems like it meets part of the definition of real communication. And these systems regulate different things in different bacteria. So this was discovered in a luminescent bacterium, a marine bacterium called Vibrio fischeri. This is a Petri plate of Vibrio fischeri. The colonies make blue light, and the blue light exposes the film. So V. fischeri only makes light when it achieves a sufficient population density. There are no biological detection systems that can see the amount of light produced by one or a few cells of Vibrio fischeri, but we can see the light produced by a crowd, like a crowd in a colony. And so as a matter of efficiency, the bacteria only make light when they're at a high enough population density so that the light can be seen by something. And we figured out how this works in the 80s. And this is a cartoon taken from a review written by my colleague Bonnie Bassler to depict what the system that we work on. So there's a gene in Vibrio fischeri called Luxi that makes a protein that's required for production of the signal. And the signal is freely diffusible out of and into cells. So when there aren't very many cells in a given environment, the signal will flow out down its concentration gradient. It can't change the concentration outside, so the concentration in the cell stays low as well. In a crowd, the signal can accumulate, and when it accumulates to nanomolar levels, it can interact with Luxar, which is a transcriptional activator, and Luxar, in its signal-bound form, will activate transcription of the luminescence genes, and all the individuals in the population make light. So this is how these systems work. This is the Vibrio fischeri signal. Steve went over the signals really quickly, but I love the signals, so in the next 2 days, you're going to be sick of me showing signal cartoons. So it's a fatty acid. It's an acylated amino acid, a fatty acid attached to an amino acid which is homoserine lactone, a non-essential amino acid. And we now know that there are lots of Luxi homologs and lots of proteobacteria, and they make different types of signals. They all have the homoserine lactone, and the specificity resides on the side chain. So I've given you this notion that, at least in Vibrio fischeri, quorum sensing controls public goods, shared resources. And so this was the example. Light production in Vibrio fischeri, no individual makes enough for biological detection, but we can see the collective activity of the group. Quorum sensing also very commonly controls exoenzymes. Again, these are public goods. All the individuals make the exoenzyme, and every member in the group can share the exoenzymes. And the idea is don't use communication so that you don't make an exoenzyme until enough can be made to make an impact on the product and get something in return. Toxic molecules are very commonly controlled by acyl HSL quorum sensing. Antibiotics, for example, again, the idea is don't waste energy making antibiotics unless there can be enough antibiotic produced to affect a competitor. There's a lot of verbiage around this one. We and others have noticed that quite often aggregation factors are controlled by quorum sensing. And I never really understood that until last summer when Ed Wilson, the father of sociobiology, published a paper, very controversial paper on how sociality evolved. And this is a line from the paper. The first step in the origin of animal eusociality is the formation of groups within a freely mixing population. And that's what aggregation does. It forces groups to form in a freely mixing population. So we have used this term quorum sensing. We introduced it in the early 90s. But what is this type of signaling really for? And there are several ideas out there, and I've just pointed out here, I'm going to tell you what the ideas are, at least the ones I thought of on the plane on the way over. These ideas are not mutually exclusive. So the first idea is that quorum sensing controls public goods, that it's a communication system for controlling cooperation. And this is surely the case in the example I gave you and some other examples. So communication is critical for cooperation. Another idea that was introduced about a decade ago is that quorum sensing signals are really so that a single individual can sense how fast things are diffusing away from it. The idea that these small molecules are very inexpensive to make, and why make a battery of exoenzymes if they're going to diffuse away really quickly? So this may be true in some circumstances, but modeling has shown that this can only happen under sort of extreme physical situations where there's very high flow, very high diffusion rates. Then there's an idea that was introduced by Burkhard Hentz, that it's all about efficiency sensing. And what he means by efficiency sensing is that it's just to be able to measure this and this together somehow. And the last thought about why bacteria do this, so this is the sum of quorum and diffusion sensing. The last idea was introduced a long time ago, and it really hasn't been studied very much, that quorum sensing enables anticipation of crowding. So that maybe bacteria can get ready for stationary phase. And this hasn't been critically tested at this point. We're gearing up to do it in Pseudomonas. It can be done in any number of bacteria. So what about Pseudomonas? This is an opportunistic pathogen that senses cell density through quorum sensing. And the idea, the glib idea, is that when Pseudomonas first invades a host, the quorum sensing signals, the stars of my show, will be at a low concentration, and there'll be little activation of whatever genes are controlled by quorum sensing. And quorum sensing controls virulence and biofilm genes, among other things. It controls a regulon of about 325 genes. And the bacteria can grow underneath the radar, and when they amass, the signal concentration, the local signal concentration will rise, and there'll be some sort of coordinated activation of the regulon. So the idea has always been, and I don't know if this is true, and I actually don't think it's true, the idea is that the bacteria can hide these virulence factors from the host until they can make enough of them all at once to overcome host defenses, and they can, in that way, deprive the host of the ability to get ready for this. So Pseudomonas uses two quorum sensing systems, and I'll call them the C4 system and the C12 system. And here's the C4 signal and the C12 signal. So C4HSL is made by a LuxI homolog called RHLI, and there's a LuxR homolog called RHLR, to which this receptor responds to this signal. Here's the C12 signal, produced by LasI, and the receptor is LasR. And I already mentioned briefly that LasR and LasI are on top of the quorum sensing signaling cascade. They regulate RelR and RelI. So in 2000, the first Pseudomonas aeruginosa genome sequence became available. This was published by some colleagues at the University of Washington. And when they showed me the sequence, there was a third LuxR-LuxI homolog that we now call QSCR. And we call it an orphan. And as I said, Vittorio likes to think of it as a solo. Here's what we know about it, or here's what we knew from the sequence. LasR and I are linked on the chromosome, they're adjacent, and RelR and I are linked on the chromosome, they're adjacent. There's no LuxI homolog next to QSCR. And this is just a alignment of QSCR, it's the second line here, and a few other LuxR homologs. And the purple here shows the region that makes the signal binding pocket. And there are certain residues that are conserved amongst all of the true LuxR homologs, and they're outlined in black here, and they're all conserved in RelI. And here's the DNA binding region. This contains a helix-turn helix domain, and there are conserved sequences, all of which are conserved in LuxR. So it looked like a bona fide LuxR homolog. And this just shows a little snippet of the genome. Here's QSCR. It's immediately upstream of one of the two phenazine biosynthesis operons in Pseudomonas, and also immediately upstream of this gene called PA1897. And I've highlighted 1897 here because it's going to become a tool for some experiments I'm going to tell you about. So orphans or solos turn out to be a rule rather than the exception, and this has been revealed by genome sequencing. EHEC has one, and Brian Ahmer will tell you about this one, right? So it seems like we ought to study them. They're all over the place. And Sudha Chugani initiated a study of QSCR and Pseudomonas, and she did what any self-respecting microbiology will be in this decade, will do in this decade. She knocked out the gene for QSCR and looked to see if there was a phenotype, and there was. This is a Petri plate of wild-type Pseudomonas aeruginosa grown on a medium that encourages phenazine pigment production, and this is the mutant. It overproduces phenazine pigments. If you make an extract, this is the wild type and this is the mutant, it makes both a blue and a red phenazine pigment at much higher levels than the wild type. phenazine production is activated by the other quorum sensing systems. So QSCR seemed to be working in opposition to LASR and RELR. So the next thing you do if you work on Pseudomonas is a virulence assay. Quorum sensing has been reported to be required for virulence. I don't know if that's true. And so you want to do an experiment to see if QSCR is involved in virulence. And we're not pathogenic bacteriologists in my lab, so we needed an easy model. And this turned out to be fortuitous for us. Another colleague at the University of Washington, Colin Manoyle, had developed a fruit fly model. And he can feed fruit flies feed them sugar water that's laced with bacteria. And if he feeds them sugar water laced with E. coli, they're just fine. If he uses a certain dose of wild-type Pseudomonas aeruginosa, then about half the flies are dead in 2 weeks. If he used a QSCR mutant, it was hypervirulence. All the flies were dead in 2 weeks. So again, it seemed to have the opposite effect on virulence that LASR and RELR have. And it was fortuitous that we used a fly model, because what I later learned was that everyone else working on quorum sensing and Pseudomonas aeruginosa was also found QSCR when the genome was sequenced. And there was at least one other group that was in the middle of some very expensive and laborious mouse models when our paper came out, and then they didn't know what to do, other than I guess they were mad at us. Okay, so what does QSCR do? It delays expression of several LASR and RELR controlled genes. It moderates virulence, and this could be from a delay in quorum sensing controlled virulence factor expression. So after what does it do, then one can ask, how does it do it? Mechanism. Well, so we supposed a few possibilities. It could sequester the acyl HSLs and thus delay activity by LASR and RELR. It could bind to the promoter regions that they normally bind to, and gene regulatory promoter regions, and occlude their binding. Or it could form heterodimers with LASR and RELR. We eventually ruled out this possibility. In fact, it will form heterodimers under certain contrived laboratory conditions, but QSCR is at much lower concentrations in cells than LASR or RELR, so that's not the way it's working in cells. So Yannick Lequette decided to use microarrays to answer these questions. And to summarize very briefly what he found, there were 31 genes that were activated by QSCR. And one has to ask, do these represent direct controls? So activated genes, none of those possibilities that I showed in the last slide can account for activation of a gene by QSCR. One of the genes that was activated was PA1897. Remember that one that I highlighted? And PA1897 is adjacent to QSCR, and we already knew from transcriptomic studies that PA1897 requires the LAS-I directed signal 3-oxo-C12HSL for induction. But Martin Schuster, when he was in my laboratory, had showed that LASR doesn't bind to the PA1897 promoter. So there was something we didn't understand. And Junhee Lee and Yannick Lequette figured this out. I'll show you one experiment that they did in recombinant E. coli. So they introduced a fusion of the PA1897 promoter and LAC-Z, and they showed that it required QSCR rather than LAS-R, and that it required the 3-oxo-C12 signal. So here are the results. If you put this fusion in an E. coli together with a LAS-R plasmid, in the absence or presence of any of these signals, there's no beta-gal, very low beta-gal activity. If you substitute a plasmid that directs the synthesis of QSCR, without any signal, basal levels of beta-gal activity. If you add 3-oxo-C12, there's a huge induction of the LAC-Z reporter, and it's specific for C12. C4 doesn't elicit a response. So QSCR requires 3-oxo-C12 HSL and activates PA1897 in E. coli. So now we have this view of an integrated AHSL circuit with LAS-R and I still on top of the quorum cascade. So LAS-I directs the synthesis of the C12 signal, and together with LAS-R, it controls a large regulon of genes that includes REL-R and REL-I. So it's dominant over the REL system, which includes a large regulon of genes. These two regulons are predominantly genes that are activated. And 3-oxo-C12 is required for QSCR activity. So it's also subservient to the LAS system. And it controls an overlapping set of genes. This set of genes is predominantly repressed, although there are these 31 genes that are activated by QSCR. And this is just to point out something that Steve Ferran sort of stressed in the last talk. So here are the three LUX-R homologs, LAS-R, QSCR, and REL-R, and they're all embedded in a regulatory network of the cell. So these things respond to signals. So a quorum is required, but that's not sufficient for induction of any specific gene in the quorum sensing regulon. There are things that regulate these, and there are lots of things that regulate individual genes in the quorum sensing regulon in addition to these LUX-R homologs. So a quorum is required but not sufficient, and it turns out that all of these 300 plus genes in Pseudomonas show different regulatory patterns from each other. So here's our orphan R homolog again, and I've shown you that there can be direct control of gene expression and that the QSCR regulon is overlapping but distinct from the LAS-R and REL-R regulons. And so now we ask the harder question, why does it do it? Well, it could enhance the capability to differentially time expression of 3-oxo-C12 controlled genes. Maybe. So I want to tell you one more thing about QSCR and then move on to LAS-R. So Junhee Lee purified it, and just to prove the point that it can directly interact with promoters that it regulates, he showed that the pure protein binds to the 1897 promoter, but it does it in an acyl HSL dependent manner. So he purified the protein. Here's a fragment of DNA and an electromobility DNA shift experiment. This has the target sequences, and this is a non-specific piece of DNA, and this is without protein added. This is with protein added and no signal. This is a high concentration of protein, and there's little or no shift of the fragment. Here's the same concentration of DNA in the presence of signal, so there's a complete shift. And here's a much lower concentration of protein in the presence of signal, so there's a shift. So there's a signal-dependent DNA binding. And I bring this up because it's in opposition to LAS-R and many other Rs. So LAS-R is thought to bind signals so tightly that we can't get it to come off the purified protein. You can purify protein, do a DNA shift, and you don't have to add extra signal. So we thought maybe this was a fundamental difference between LAS-R and QSCR, that LAS-R responses would be committed. If you take signal away, the LAS-R response will continue until the protein is degraded. If you take signal away, QSCR responses will stop immediately. So it has also been thought, as Steve alluded to, that LAS-R and TRA-R require cognate signal to fold into an active conformation. And once they fold around the signal can't come off. And we thought QSCR was different from these purification studies.
What about binding, the box? The QSCR is standard Lux box?
It is, yeah. There are a couple of nucleotides that are specific for QSCR binding versus LAS-R binding.
So there's no cross, so LAS-R cannot activate the QSCR regularly?
That's right. In our recombinant bacteria, there's no crosstalk. What happens in Pseudomonas with all of the assorted Lux boxes that we know about, we're not clear about that.
So you can purify QSCR without signal in a soluble form.
So the question was, can you, I'm going to rephrase that into a question.
Feel free, yeah.
Can you purify QSCR without signal in a soluble form? And the answer is no. That's what we thought from this experiment. Well, Steve remembers the story now, but he asked the question, yeah, so I'm going to answer it.
I'm old.
I'm old too. So we were surprised to find that our purified QSCR had bound signal. There was a lot of signal. And it turns out that during our purification, the concentration of QSCR was always at micromolar or higher amounts. The affinity for signal is nanomolar. So if you put QSCR in solution at micromolar amounts, you can find free signal in the environment, but it will be at micromolar amounts because QSCR retains signal. And that's a sufficient amount of signal so that just equilibrium chemistry tells you that all the QSCR will be bound to signal. So we always keep it in solutions at high concentration, but when we do our gel shift assays, we dilute it out to sub-nanomolar concentrations. And during the gel shift assays, the signal diffuses off of QSCR, and the only way we can get a shift is to add signal. So it took us about 10 years to figure that out. And I explained it to Steve Ferrand once, and he got it, but he already forgot it. So let's see. So that's a clue that there's something wrong with the story. We were thinking that reversible binding as opposed to tight binding by LAS-R might represent a fundamental difference. And I already told you this. Once activation is commenced with LAS-R, the response should be committed until the LAS-R protein is degraded. So we wanted to test this. So here's the model. The TRA-R, so it was seeming to appear that there were 3 classes of LUX-R homologs. Most of them were like TRA-R and LAS-R. And the idea is that as the nascent polypeptide is coming off the ribosome In order to fold into an active conformation, it has to fold around the signal. And then once bound to signal, the signal doesn't come off. So it's an irreversible reaction. We had studied Luxar, and we thought that it also required signal to fold into an active conformation. Signal binding was reversible, but this form of the protein, once purified, is very unstable. And we knew that ESAR, which is a repressor from Pantoia, I guess it's called now, you guys are the plant guys, was different. And there's another example of one that works like this. It doesn't require signal to fold, and signal binding is reversible. So there were these three ideas. With LASR and QSCR, we wanted to test the reversible versus irreversible hypothesis, and we got a big surprise. The basic approach is simple. We wanted to ask about the response to a rapid signal withdrawal. And this question of the response to a loss of signal has been barely touched over the years. I've given you the two references I know about on what happens when you take signal away from a Luxar family member. We published a paper in 1985 and Steve Ferran in 2008 on TRA-R. Almost all of the work that you can find has been on a response to either the natural accumulation of signal or an exogenous addition of signal. What are the signals activating? So the experiments are really simple. You can grow up cells in the presence of signal. We used I mutants, centrifuge them out of their culture medium, and resuspend them in signal-free medium. So that's what we did. And we found that LASR is not a committed event. And these two panels show an experiment done in recombinant E. coli and an experiment done in Pseudomonas aeruginosa. So here, this is expressing LASR and a reporter that responds to LASR. And if you grow a control, you see an induction here. If at the arrow, you centrifuge the cells out of the medium containing signal and resuspend them in a medium containing signal, the response continues on in a fashion similar to the control. If you resuspend them in a medium without signal, there's no further response. So the LASR response seems to be, this would suggest that it's reversible, that signal is coming off and LASR activity ceases immediately. And this just shows the same thing in Pseudomonas. It's true in Pseudomonas too. These inserts are time courses over the first period after the signal is taken away to show that LASR activity is actually going down over time. So will LASR that's free, so we're supposing that we've made a pool of LASR that's free of 3-oxo-C12 because it's diffused off. And we wanted to know whether this LASR will retain the capacity to bind 3-oxo-C12 HSL if we add it, really to push the idea that maybe there is reversible signal binding. So here we had LASR under control of the arabinose promoter. And if you grow control cells and centrifuge them out at the arrow and resuspend them in the presence of arabinose and 3-oxo-C12, you get a continued response. And we made this promoter very weak so that there's essentially no promoter activity, no LASR promoter activity in the absence of arabinose. So if you centrifuge the cells out here and don't add arabinose back, but add 3-oxo-C12 back, the response continues on. And it sort of levels off here because there's no new LAS-R being made. If you wait 20 minutes before you add the signal back, you still get a response. So that means there must have been some pool of stable signal-free LAS-R. Even if you wait an hour, there's a response, a little bit of a response. This insert just shows that the arabinose promoter really did effectively regulate LAS-R transcription. Here is LAS-R transcription right after removal of arabinose, or LAS-R transcript levels right after removal of arabinose and 20 minutes later. The half-life of the LAS-R transcript is 1.5 minutes. So we had this idea that there was a pool of LAS-R that was existing in cells that maybe, I haven't shown you all the experiments, can fold in the absence of signal and is stable in the absence of signal. And to really try and prove the point, we did DNA mobility shifts with extracts that were grown with 3-oxo-C12 or without 3-oxo-C12. And the trick of this experiment was to break the cells into a cold buffer that had 3-oxo-C12. So we had learned that all of these LuxR proteins, if you try and get the signal off, they're very unstable in the absence of signal. So maybe you can't show active LAS-R in cell extracts of cells grown without 3-oxo-C12 because it's unstable after you break the cells open. So to try and stabilize it, we added 3-oxo-C12. So here are results with cells that were grown in the presence of 3-oxo-C12 HSL. And this is, again, a gel shift experiment with increasing, let's see, how did we do? I can't remember how we did this experiment. Oh yeah, increasing amounts of protein. So even at pretty low protein concentrations, there's a gel shift that one can observe. So these are actually 10-fold dilutions of the protein. And here are cells grown without 3-oxo-C12, but broken into buffer that contains 3-oxo-C12. Again, at high concentrations of proteins, there's a specific DNA shift. So there was some active LAS-R in here. And there's not as much as in cells grown with 3-oxo-C12, about 10 to 50 times less, but still there's active LAS-R that's produced in these cells without signal. So we, and by we I mean some collaborators I have at Vertex Pharmaceuticals and about 50 other groups around the world, maybe some of you in this room, are interested in quorum sensing interference as an antivirulence therapeutic. And because of the ease in setting up assays, most of the groups have targeted LAS-R. But we always wondered how this was going to work if LAS-R was folding around its ligand as it was produced and then it didn't let go. So how will adding a therapeutic work on an active protein? We'd have to ask a lot of a small molecule therapeutic. But I'm warming back up to this idea because it now seems as though this is the model that's correct, at least for LAS-R and maybe a number of these other LAS-R homologs. And so the data we have now suggests that the way these things fold, at least LAS-R and QSCR, is they fold into an active conformation in the absence of signal. If there's no signal around, they're targeted for proteolysis. So they have a short half-life. There's a small stable pool of LAS-R in cells. It can be stabilized by binding to the cognate signal, which also pushes the equilibrium towards dimers. And the dimers are the active form that can bind target DNA. So the acyl HSLs just push the equilibrium in this direction. In the absence of acyl HSLs, the equilibrium goes this way and targets these proteins for proteolysis. So what I've tried to do this morning is give you a bunch of background information and then take you through to what's our current thinking on LASR and how it functions. And I just want to, so these are my collaborators at Vertex Pharmaceuticals. Uda Mu, she's the essence of the corporate person here, and Brian Hanzelka. And I talked about some of the work that Martin Schuster did as I went on this morning. He's now just becoming an associate professor at Oregon State University. And this is my lab in its current incarnation. And this is for Giordano, who spent some time in my lab. And he may recognize a few of these people, but there are a lot of different people now. And I'd be happy to answer any questions you have.